Thank you. I'm going to talk about the use of topological data analysis in the context of visualizing biomolecules. Topology here refers to the study of shape and the connectivity of space. In the recent few years, it's kind of accelerated, but started off about 15, 20 years ago. There is, for close to 2 decades, there's been a concerted effort between mathematicians who are interested in topology, theoretical computer scientists, and a good number of visualization researchers and also researchers in other domains. Such as geometry processing and computer graphics, who have come together to develop several techniques that help us understand connectivity of space and have designed computational tools. In the past 10 years or so, these have been very successfully used for visualizing biomolecules. My name is Vijay Natarajan, so I work at the Indian Institute of Science. So in today's talk, I'm going to use some illustrations based on our work to give you a flavor of how topology has played an important role in the development of biomolecular visualization tools. So this work is definitely thanks to the kind of inspired efforts of various folks. I want to acknowledge them here initially. I want to highlight specifically past students Talha, Nitin, and Dilip, and current PhD student Raghavendra, who have done a lot of the work that I'm going to talk about today. So let's begin by talking about proteins, which is what I mean by when I say biomolecules in this talk. Proteins play an important role in in building living systems, right? So and it's well accepted that the structure of a protein plays an important role in its function. And for those of us who are studying or developing geometric algorithms, this is good to know that it's already well accepted that structure plays an important role in the study of proteins, so we can directly use the techniques we know. Cartoons are a good way to visualize protein structure. However, there are several other representations of biomolecules. Each comes with its own set of advantages. As an example, space-filling models that you see on the right is also quite popular as a way of both visualizing a biomolecule, but in addition, it also helps compute several measures such as volume and surface area of the molecule. So in addition to such space-fill models, there are several other representations, such as the surface representations that help understand how one biomolecule or a protein interacts with another. And among the long list of representations, there are voids, which help, among other things, study the stability of a protein. In this talk, I'm going to discuss one specific representation that falls under the category of space fill called alpha shape. And some of the surfaces that I show later are what are called the molecular skin surface. So why geometry? Why a geometric representation of biomolecules? The obvious answer, the first answer is, like I said, it helps capture various geometric measures such as volume, surface area, and so on. But in addition, it plays an important role in several simulation and computational tasks, such as ab initio modeling, molecular dynamics, the study of how drugs bind to various active sites on proteins, and the study of protein-protein interactions. So let me jump in by talking about one representation, so we have a sense of the kind of geometry that plays a role here, and then move on to topology. The illustrations I'm going to use are in 2D. So let's start with a set of weighted points. Let me call it S. So this is a collection of points P sub I on the plane, together with a weight R sub I. One way to think about these weighted points is think of each weighted point as a disk, where the radius R sub I is the radius of the disk, and the point P sub I is the center of the disk. So here on the picture here, what you see is a collection of disks. We want to talk about the union of these disks, because this fills some space, and we want to talk about that space. Now, in order to do that, I need a construction called the Voronoi diagram. Let me talk about that briefly. Given a point on the plane P, I want to talk about how far it is from each of these disks. And the way I define the distance is different from Euclidean distance. So this is called the power distance between a point P and one of these weighted points P sub I. This is essentially the square of the distance between P and the center of the disk, together with a subtractive weight, which is equal to the square of the radius, right? Now, with this distance, one can talk about the regions associated with every weighted point. So let's say I talk about this one weighted point that I'm highlighting here. The region associated with this disk is the collection of all points that are closer to this disk as compared to other disks, right? So it's kind of each disk saying this is my territory. So this region, as you can see, is bounded by these green line segments. One can show that these regions are always convex, and each disk has a region for itself. And this decomposition of the plane into such regions is called the weighted Voronoi diagram. If there are no weights, then we call it the simple Voronoi diagram. The connectivity between the regions or the cells in the Voronoi diagram can be captured in this dual structure consisting of the nodes which come from the P sub i's and the edges when you have two regions that are adjacent to each other. And this dual structure is called the weighted Delaunay triangulation. So there is really no difference between the Voronoi diagram and the Delaunay triangulation, except that these are two different representations of the same construct. And we use one over instead of the other because it's more convenient. So far, we have talked about the representation of the entire plane. The Voronoi diagram or the weighted Voronoi diagram decomposes the entire or partitions the entire plane. We are, of course, interested in the region occupied by the disks. In order to capture this, we look at a substructure, and this is called the alpha complex. And in order to reach there, we talk about a growth model where the radius of each disk is grown in the following way. You increase it by you increase the square of the radius by a value alpha, and this is the alpha that refers to the parameter in this complex, the alpha complex. Now, the alpha complex at alpha equal to 0, as you can see here, is shown in red, captures the connectivity of the disks. The union of these disks is connected, has a single component. So does the red complex that you see here. The union of disks has a tunnel, and so does the alpha complex. So the alpha complex is is a good representation. It's a representation so far. We haven't seen why it's good. It's a representation of the connectivity of this union of disks. So now we can throw away this geometric representation, um, uh, looking at the disks and so on, and instead work with something which is more amenable to computation, namely the alpha complex. The alpha complex is essentially a collection of nodes, edges. Edges represent the intersection between two disks, and triangles represent the intersection between three disks. The growth model helps us capture the size of these tunnels and voids. So these are features of interest to us. So let's see an example here. When you increase the alpha to alpha sub I, the disks overlap, and they occupy the empty space. And this closes one of the tunnels. However, you notice that it actually seems to have created another tunnel, right? Um, so, so there is clearly some value in the growth model, but how does it help capture these various tunnels and at different scales? In order to see that, we want to look at not just a specific alpha, but the entire sequence of alpha complexes for varying values of alpha, right from 0 till infinity. Now, when we do that, as you see on the pictures on the top, where you go from here for low values of alpha, I'm not showing the disks now. I'm just showing the edges that come in when two disks intersect. And as you go increase alpha, you notice that there are more edges that get introduced. And at some points of time, you have cycles. So the alpha complex helps us represent the topology of the union of disks. But now we want to talk about the representation of these tunnels and components, which are features of interest to us. At some instance during this growth, we have a creation of, or a birth of, a cycle. And at some later instant of time, so in this instant, at this time instant, for example, this small cycle is created, and at a later instant, the cycle is filled because the triangles come in and fill the tunnel. So the tunnels have a lifetime, which is represented by the birth and death time instant. So this birth-death pair is called the persistence pair. And there is much algebra that helps represent these birth-death pairs. In fact, these are called homology groups. The zeroth homology, shown here in red, with the x-axis showing the evolution in terms of time or the growth, the alpha parameter, if you will. The red shows the evolution of the connected components. And so you see that at the end of time, at infinity, there is still one connected component that survives. The green shows the birth and death of tunnels, one-dimensional cycles. And the length of these intervals, the green intervals, show the lifetime of these tunnels. And what you see down below is the two-dimensional tunnels, which are voids. So this notion of topological persistence, there is much algebra and topology hiding in the background here. They help us measure the size of components, cycles, and voids. Coming back to proteins, the study of cavities helps us understand how one protein interacts with another and with other ligands. The study of tunnels helps us understand transport mechanism of ions through proteins, especially if they are lying on the cell membrane. The alpha complex helps us both represent and compute these features of interest. Going back to the set of disks, a pocket or a void can be captured by studying the complementary space, this the complement to the space occupied by the union of disks. The tunnel and the pore can be represented. The pore is the channel through which ions could pass through. These can also be represented by studying the complementary space. A simple algorithm that computes these features of interest would recognize a potential binding site or and the entry region into the protein, and construct by studying the complementary space that comes from the alpha complex. It would construct an explicit representation of the tunnel that leads us towards the binding site. Now, this is a very rich representation. It allows us to visualize and understand the path towards the binding site, but in addition, it helps us understand the energetics along the path. So we have used this idea to compute and to visualize biomolecular channels. So this is, like I said, just an illustration. There are several such studies within the visualization community and within the bioinformatics communities. The pipeline is essentially what I said. We start with the volume representation, compute the alpha complex, and eventually compute the pores. Of course, there are several other components to handle noise and to identify significant, biologically significant pores and so on. This can be used to compute transmembrane pores and also pores in other molecules. The tool that implements this algorithm is also available and has been used towards the study of several transmembrane pores by other groups. So once you have a good representation of the protein molecule and its complementary space, it supports several related computations. For example, it helps identify, even in the presence of noise, whether you actually have a cavity or a transmembrane or a pore, which could be a transmembrane pore. So in this case, by incorporating a good model for noise and extending the alpha complex based algorithm that I talked about earlier to become robust even in the presence of noise, one can connect what seem to be cavities into a single pore. So we showed that there are several channels which could not be detected unless you allow for such connections between cavities and allow for the incorporation of or acknowledge the presence of noise in your data. In addition to space filling models, surface representations have also been quite effective in the study of biomolecules. So surface representations again are geometric, but topological constructs such as what is called the Morse-Smale complex, which is similar to the flow-based topology that Ingrid talked about, have played an important role in studying how one surface aligns with another. And finally, moving away from discrete representations where you have a bunch of atoms forming a molecule, biomolecules can also be studied via imaging and studies of scalar fields such as electrostatic potential, hydrophobicity, and so on. And in this context, topological techniques can be used to identify symmetry or repeating patterns within the molecule. There is a nice topological structure called the contour tree that has been used to identify these repeating patterns. For example, in this human adenovirus, which has dodecahedral symmetry, and the symmetric patterns have been identified and highlighted in this volume rendering. And again, providing tools helps others use this technology and put it to test. So I want to end by saying that geometry plays an important role in understanding protein shape. And we are happy that this is a well-accepted fact already within the bioinformatics and the structural biology worlds. But what we also find is that good representation of features and efficient computation using these features require a sufficiently sophisticated topological representation. So what we need in future is more integrated representations that use topology together with traditional representations of biological structure and techniques for interacting with such representations. So I will end there, and I guess we are ready for questions.
Thank you very much, Vijay. Very nice talk. So I believe we have, for now, we have one question in Discord. That is, Alexander Reed asked, I didn't quite get why you'd use weighted instead of normal Voronoi diagrams. Could you explain again what benefits this complication brings?
Okay, that's a good question. So let's talk in the context of biomolecules. Each atom comes with its own radius, and hence we don't want to think of the points as uniform. Rather, they come with their own weight, and hence the use of the weighted Voronoi diagram.
All right, thank you very much. Then one more question: is this method good in predicting protein active sites?
Okay, so that's a good question. So analysis, I like to distinguish analysis from prediction, and we often are very conservative when we say anything here. So I would say that our techniques are good for analysis, the techniques I presented here, but it requires sufficient additional information in order to predict active sites. For example, these techniques together with a database of known active sites for similar molecules could be used towards a prediction tool. Right. So, yes.
All right, then thank you very much. And also thank you, Ingrid, again. I saw that Ingrid answered all of her questions in Discord already.
Great.
If there are more questions, they'll pop up in Discord, so maybe you can take a look later. If not, thank you guys again. And I think we'll now enter a 15-minute break and meet back here at 3:15 for our next talks.
Okay, thank you very much. Have a
Thanks.
good day.